All right. Previously, we learned about the history of the discovery that DNA is the genetic material and also the structure of DNA, who figured out the structure of DNA. And it turns out once the structure was discovered, then that allowed us to figure out how it functions. So yeah, it is a long, twisty, and a rope laddery-like thing, and it's and we are going to use that analogy of a ladder because that is a good analogy. It looks like a twisted ladder, a double helix, a twisted ladder, like we're seeing here in the animation. But as you can see, there are a lot of components that come together to make up DNA. So we're going to take a look at the details of those components so that we can better understand how DNA functions. So let's start just with the name, DNA, deoxyribonucleic acid. Deoxyribonucleic is all one word, and then acid, deoxyribonucleic acid. Let's start with the cell, and this is basically a review of what we've learned previously. And you know that this cell, because it has a nucleus, is a eukaryotic cell. And if we take a look at what might be inside this eukaryotic nucleus at the time, just leading up to cell division, we would find chromosomes, and they would be condensed and made up of two sister chromatids. Therefore, they would be mitotic chromosomes because the cell would be going through the process of mitosis. If we unravel that chromosome, we'll find that what it's made up of is a combination of DNA and proteins called histones that the DNA is wrapped around and then packed together. That's how it condenses into mitotic chromosomes. But if you'll recall, that combination of DNA and protein is what we call chromatin, which is DNA wrapped around clusters of histones, which are proteins, and there are 8 histones in each nucleosome that DNA wraps around. But what we're going to do is now focus in on what makes up just the DNA by itself. Not chromatin, not histones, just the DNA itself. That's what we're going to spend most of our time looking at the structure of. And as we follow down the diagram here, we see that the DNA unwinds and unzips. Well, it doesn't unzip, actually. It unwinds, and we can see clearly that there are 2 polynucleotides. If you'll recall, nucleic acids are organic biological compounds, and most organic biological compounds are macromolecules because they are polymers. They're long chains of repeating units that we call monomers. And in the case of DNA, the monomer that links together into long chains to form a polynucleotide is a nucleotide. And here along, if we use that ladder analogy, here along the rails of the ladder, which is also referred to as the backbone of a polynucleotide, we see that there are alternating sugar, phosphate, sugar, phosphate, sugar, phosphate molecules. And that's the same in all DNA molecules. That doesn't change. The sugar is deoxyribose. That's where the deoxyribo of deoxyribonucleic acid comes from. And then, of course, there's phosphate, which is the same as the phosphate that we saw in ATP. Back when we were studying ATP, remember the TP stands for triphosphate. And then again, the deoxyribose and phosphate alternating makes up the rails of the ladder, also known as the backbone of the polynucleotide. And then you can clearly see on the interior of the molecule are these nitrogenous bases. Guanine, cytosine, adenine, and thymine. They are known, just generally referred to as bases. More specifically, they're nitrogenous bases because they contain a lot of nitrogen. But they're not only bases, they are nucleotides. In other words, the different nucleotides, there are 4 different nucleotides because there are 4 different bases. Each nucleotide has a different base. Each of the 4 different nucleotides, different base. And these 4 nucleotides pair up in 2 different pairings. So there are 2 nucleotide base pairs. So if you look at the diagram here, there's adenine and thymine that pair up, and there's guanine and cytosine that pair up. In other words, AT pair up, A and T pair up, and G and C pair up. And that's why it's useful to remember these nitrogenous bases or these nucleotides in the order ATGC, adenine, thymine, guanine, cytosine, because that way it helps you remember which ones pair up. And here's the significance of these different nucleotides and bases. It's the sequence of these bases, the A's and T's and G's and C's, that spells out the information in a gene. The sequence of nucleotides codes for a protein. That's what a gene is. It's a sequence of nucleotides that codes for a protein. If we come back to the structure here of the DNA molecule, you'll notice that these two polynucleotides are held together by hydrogen bonds. And there are 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between guanine and cytosine. That's part of the reason that A will only bind with T and G will only bind with C. It's because of their structure and because they want to form a certain number of hydrogen bonds between them. How do we know all this? Well, it was finally figured out. All the pieces of the puzzle came together and were put together. And literally, pieces of the puzzle. They built this model out of cardboard and tin by James Watson and Francis Crick. But remember, a lot of people came before them, first to discover that DNA is the genetic material, and then Chargaff and Franklin contributed very much to Watson and Crick being able to figure out this final model of the DNA molecule. So to summarize what we learned on the previous slide, the DNA consists of 2 polynucleotides that are joined into a twisted ladder-like structure called a double helix. A molecule of DNA, which is an unduplicated chromosome or duplicated chromatid, is made up of millions of monomers called nucleotides. And each nucleotide itself consists of 3 different parts. Each nucleotide consists of a phosphate group, a deoxyribose sugar. Notice it ends in OSE. It's a pentose sugar because pent refers to 5. And a nitrogenous base. So each nucleotide has these 3 components: phosphate, deoxyribose, nitrogenous base in a DNA molecule. And of course the nitrogenous base is either adenine, guanine, thymine, or cytosine. And I said them in the wrong order. I should have said adenine, thymine, guanine, and cytosine, because adenine and thymine bind together and guanine and cytosine bind together. But this is what these nitrogenous bases is what makes different nucleotides different. And it's, again, the sequence of nucleotides that spells out the information in DNA, that spells out the information of a gene that then can be used to make a protein. Now, if you'll recall, DNA is not the only nucleic acid. There's also RNA. And we're going to be looking at RNA in great detail also and see how RNA functions in the process of making proteins. But that's for a later discussion. Let's just take a look at what a nucleotide looks like. So this represents a nucleotide. So again, it's a phosphate, a pentose sugar, a nitrogenous base. That's one nucleotide, one DNA nucleotide. If the sugar is deoxyribose, it's a DNA nucleotide. If the sugar is ribose, it's an RNA nucleotide. And again, these nitrogenous bases are going to be adenine, thymine, guanine, or cytosine in DNA. If this is RNA, however, there is no thymine. Instead of thymine in RNA, there's uracil. But it functions the same way as thymine. In other words, adenine and thymine bind together in DNA. Adenine and uracil bind together in RNA. And then finally, I just want to relate something that we learned in the past that is related to this because it's another kind of nucleic acid. So the 2 that most people think of when they think of nucleic acids are DNA and RNA. But there's this 3rd one that we've learned about previously. And so this is the question that you're being asked, and you can pause it and think about it for a minute, but I'm going to give you one more clue. It's the energy currency of all living cells. What is that? The energy currency of all living cells is made up of ribose, the sugar ribose, the nitrogenous base adenine, and then 3 phosphate groups. Instead of just 1 phosphate group like we see in a nucleotide, there are 3 phosphate groups. If you recall, that's adenosine triphosphate, ATP. So ATP is also a nucleic acid. I just wanted to remind you of that. So here again we see the nitrogenous bases. We're going to go ahead and list them here again just for practice. I know it's getting redundant, but it's good for you to write these things and think about them over and over and over again so you remember them. So A, of course, stands for adenine, T, thymine, G, guanine, C, cytosine. And we also see on this diagram that there are 2 classifications for these nitrogenous bases. There are nitrogenous bases that are made up of 2 rings, and that would be adenine and guanine, and those are the ones we call purines. Purines are the nitrogenous bases that have 2 rings, and then thymine and cytosine are nitrogenous bases that are only made up of 1 ring, and those are known as pyrimidines. And the way that I remember that, which is a purine and which is a pyrimidine, and how that relates to the number of rings, is the shorter word has 2 rings, is the larger molecule, and the longer word is the smaller molecule with only 1 ring. At least that's how I remember it. So if we answer these questions, what makes them different from each other? It's the structure. Their structure is what makes them different from each other. And then can you infer from the shapes of the nitrogenous bases? Well, and what it's asking about are the shapes of these symbols of nitrogenous bases. So you'll notice that they're kind of shaped like puzzle pieces. And as you know, puzzle pieces fit together, which is what we're going to see on the next slide. So here we're looking at the base pairing rule and why it is that certain bases will only bind with other bases. Let's also look at the bonds that hold the nucleotides together, and also what bonds the nucleotides together to form a polynucleotide, in other words, along the backbone. So all of these bonds are covalent bonds. So it's a covalent bond that bonds the nitrogenous bases to the deoxyribose. But do note that that's what the nitrogenous bases bond to, is the deoxyribose. And then when it comes to nucleotides bonding together to form a polynucleotide, it's between deoxyriboses, it's between sugars. But remember, what's between sugars is a phosphate. And this is a covalent bond, but it's got a special name because it's 2 bonds with a phosphate in between. 2 bonds with a phosphate in between is what holds nucleotides together in a polynucleotide. So it's known as a phosphodiester bond. Phospho for the phosphate, di for 2, and then these are ester bonds, but we're not going to get into the chemistry of what makes them ester bonds. But that's what holds nucleotides together in a polynucleotide, phosphodiester bonds. All the other bonds are covalent bonds, except for the bonds between nitrogenous bases. So here we have adenine, thymine, and they will only bond with each other because one is a purine and the other is a pyrimidine. And it's the same for guanine and cytosine too. One's a purine, one's a pyrimidine. So a purine always bonds with a pyrimidine. So it's because of their structure, but it's also because of their hydrogen bonds, the number of hydrogen bonds that they want to form. So there's only 2 hydrogen bonds between adenine and thymine. So that's a base pair. And then we've got cytosine and guanine, that's also a base pair, one purine with 1 pyrimidine. But they want to form 3 hydrogen bonds. So again, it's a combination of their structure. So each base will only bond with one other specific base, and that's what makes it a rule. Will only bond with one other specific base due to their structure and the number of hydrogen bonds. And if you'll recall, what makes this really significant is the hydrogen bonds are the weakest kind of bonds that we've learned about. And as far as I know, they're the weakest kind of bonds there are. So that means that it's very easy for the DNA, the two polynucleotides that make up DNA, to separate, to come apart, what we're going to refer to as unzipping. And that turns out to be a crucial part of the function of DNA, because in order to replicate, it has to unzip. The two polynucleotides have to unzip. And in order for RNA to be transcribed from DNA, the two polynucleotides have to unzip. So it's a crucial part of the structure that the two polynucleotides are held together between nitrogenous bases by hydrogen bonds, because they're weak and they can come apart relatively easily. Also notice that these polynucleotides are upside down to each other. That's known as being anti-parallel. The two polynucleotides that make up the double helix are anti-parallel to each other. They're upside down relative to each other. And there are names for the ends of the molecule, and they're named for the number of carbons on the deoxyribose. So there's a carbon number 5 at this end, and there's carbon number 3 at this end. This is carbon number 3 right here, and carbon number 5 is actually outside the ring of the molecule here, but it's not shown in the diagram. But if we look over here, so from top to bottom here, this is what we would call 5' to 3' is going from top to bottom. But on the other side, from top to bottom is 3' to 5'. So they're upside down relative to each other. They're anti-parallel. That's what we call anti-parallel. All right, because DNA is made up of polynucleotides, that means they have to polymerize. In order to build a polynucleotide, you have to string nucleotides together and do a chain, and that's called polymerization. So we're looking at nucleotide polymerization here, and we're building a DNA molecule from scratch. So this is going to give you a lot of practice. The whole reason there's empty space on this slide is so you can draw in your own DNA molecule built from scratch. And we're going to start with deoxyribose and phosphate. Remember, that's what makes up the backbone of the molecule. But bound to the deoxyribose is one of these nitrogenous bases. And we're just going to go ahead and start with A here, adenine. So this, together, if you'll recall, represents one nucleotide. So that's one DNA nucleotide. And again, it's DNA because it has deoxyribose. If this was ribose, it would be an RNA nucleotide. Next, we're going to go ahead and construct one of the backbones of one of the polynucleotides. So all that means is that, and this is what you should do too, is you're alternating deoxyribose phosphate. In other words, DP, 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 all the way along here, just like this. And remember, you can pause if you need time to do that. And again, what this represents, if we're using the ladder analogy, like we are here, this represents what we're going to refer to as the rails of the ladder. But you could also refer to it as the backbone of a polynucleotide. So it's either the rails of the ladder or the backbone of the polynucleotide is made up of alternating deoxyribose phosphate, all along, and that part of the molecule, that part of the polynucleotides is the same in all DNA molecules. What's different in all DNA molecules is the sequence of nitrogenous bases that are associated with each of these nucleotides. So I'm just going to go ahead and put up this sequence, and you should copy it. But then after you copy it, go ahead and match it up with, according to the base pairing rule, match it up with the nucleotides that would be opposite each one of these nitrogenous bases. So in other words, A matches up with T, T matches up with A, G matches up with C. And I really meant for you to practice that before I pulled it up, but too late now. So the point is that these base pairs, as we call them, each one of these is a base pair. And by the way, DNA, the length of DNA is often described in base pairs and can be millions of base pairs long. And as we add the other rail to the ladder, or the other backbone to the other polynucleotide, we see that the nitrogenous bases, the base pairs, are what form the rungs of the ladder. So the deoxyribose and phosphate form the backbone, or the rails of the ladder, and the nitrogenous bases form the rungs. Now, the way we built this is not the way that DNA is actually built. It's actually polymerized. The way it's actually polymerized is one nucleotide at a time. So deoxyribose phosphate and a nitrogenous base is what makes up a nucleotide. So one nucleotide at a time would be added to form the polynucleotides that makes up DNA. And you'll see how that works when we do our lab, where you're going to be both replicating DNA and also transcribing RNA from DNA. Both of those involve nucleotides being strung together into polynucleotides. So here's another representation of DNA, and we're going to use it just so you can practice some more, the base pairing rule, just to make sure you got it down, just to make sure you don't, you're not going to make any mistakes if you're ever asked to do it. So in this diagram, nucleotides are represented here by deoxyribose being this blue rectangle, and the phosphate being the lighter blue rectangles, and then the nitrogenous bases being the different colors. So that's one nucleotide. This represents one nucleotide, a little different than what we saw on the previous slide, but not much. And then, of course, adenine binds with, you know it, thymine, and guanine binds with, yeah, cytosine. And remember, there are different numbers of hydrogen bonds between them. There are 2 between adenine and thymine, and 3 between guanine and cytosine. And that's partly why they will only bond with their partner. In other words, according to the base pairing rule, it's because one is a pyrimidine, one is a purine, and one's a pyrimidine. That's one reason. The other reason, and that if you take that further, that explains why they want to form different hydrogen bonds, different numbers of hydrogen bonds between them. You'll also note in the diagram, these dots here are representing the hydrogen bonds, but there's kind of a mistake in the diagram because they show correctly show 2 between adenine and thymine here, and 3 between guanine and cytosine, but then here they make a mistake and show 3 between adenine and thymine, and here only 2 between guanine and cytosine. So they're not being very consistent in this diagram, unfortunately. They're not being very consistent and correct. But for right now, go ahead and practice. Go ahead and practice putting the complementary, and that's the term we use to describe that. A is complementary to T, G is complementary to C. So go ahead and put in the complementary nitrogenous bases that match up on this polynucleotide that is going to be complementary to the polynucleotide that's already labeled. And notice that because this is a helix and these 2 polynucleotides are wrapped around each other, this is the way that this polynucleotide follows in the diagram. And then the one you're filling in follows this way. All right, so if you need to, go ahead and pause it and go ahead and fill those in. Okay, so now we can go ahead and check your work. So adenine binds with thymine, cytosine with guanine, adenine with thymine, guanine with cytosine, guanine with cytosine, adenine with thymine, guanine with cytosine, cytosine with guanine, thymine with adenine, and guanine with cytosine, finally. So again, the fact that one polynucleotide matches up with the other polynucleotide is the whole idea of complementarity. That polynucleotides are complementary to each other, and the nitrogenous bases are complementary to each other, and you can say the nucleotides are complementary to each other. And all that means is that the nucleotide sequence of one strand determines the sequence of the opposite strand. They have to match up. In other words, they have to be complementary. They have to match up according to the base pairing rule. So here's even more practice, just to make sure you have this down completely. Go ahead and complete the polynucleotide in this DNA molecule, and go ahead and complete the complementary sequence for this sequence of nitrogenous bases or nucleotides. Go ahead and pause it and fill those in. All right, let's check. Of course, A matches up with T, C with G, T with A, C with G, A with T, G with C, T with A. And then if we do the complementary sequence for this sequence, A matches up with T, T with A with T, T with A, C with G, A with T, T with A, G with C, C with G, G with C, G with C, and of course G with C. And I'm sure you all got that correct, because it's not that hard. All you got to do is remember A, T, G, C. And then let's finish up with an analogy for the genetic code. And the analogy is that the genetic code is like, each individual letter, or the nitrogenous bases, or the nucleotides. But when you string them together, they make words. And the words make sentences and paragraphs, and the sentences and paragraphs come together in a book. All right? So the nitrogenous, and remember there are four. The four nitrogenous bases are like a four-letter alphabet. And it's amazing that all the information to spell out every living thing on the planet is spelled out with only 4 letters. That's incredible. It's the sequence of those bases. And they're read in triplets. In other words, 3-letter words. The analogy here is that these codons are like 3-letter words. Each codon codes for one amino acid in a polypeptide or protein. That's where we're going next. Next, we're going to go and take a look at, after we look at replication, how DNA copies itself, we're going to take a look at how amino acids are strung together, how the information in DNA is transferred to RNA, and then RNA strings together amino acids into polypeptides and proteins. That process begins with the process of transcription. So this is your introduction, at least in this course, to how DNA is transcribed into RNA. And it works just like we were just doing, where you were matching up A and T and G and C. But when it comes to RNA, if you'll recall, there is no T in RNA. There is no thymine in RNA. It's replaced by U. It's replaced by uracil. So adenine does not match up with thymine. It matches up with U, uracil. And then G matches up with C, just like it always does, and C matches up with G. T matches up with A. There is adenine in RNA. So all of this is exactly the same, except for when it's time to make RNA from DNA, and if the DNA has adenine, that means the RNA gets uracil. Uracil matches up with adenine, just like thymine does in DNA, but RNA doesn't have thymine, it has uracil instead. So anytime that there is an A, you need to remember that it's not T that matches up with A. If you're transcribing RNA from the DNA sequence, that means that you need to use uracil, U, instead of T. All right? U is for uracil. If we continue with the analogy, words form sentences and paragraphs, which are genes, and the result is an infinite number of books. In other words, species genomes. Each book represents the genome of a species, all the genes that make a species a species. Like when we talk about the human genome, we're talking about all the genes that make a human a human. All right? So the central dogma of biology is what's being shown in the diagram below. It's everything from the sequence of nucleotides in DNA, which spells out the genes, those genes then being transcribed into RNA. The RNA then, and there are 3 different kinds of RNA that work together to translate proteins, and then the proteins are what lead to the phenotype of an individual. All right, so everything from gene to protein, the whole process of going from gene transcribed to RNA, translated into protein, and then the proteins produce the phenotype. The proteins run the show. All that is referred to as the central dogma of biology, and it's the essence of life. It's the way that every living thing works. So to run through that again, a gene is a section of DNA that codes for a protein. It's a sequence of DNA. You could also say it's a sequence of DNA that codes for a protein, and that would actually be a better way to say it. This is known as the one gene, one protein hypothesis, which is generally how it works. There's a little more to it than that, but for our purposes, that works great. One gene codes for one protein. Each gene has a unique sequence of bases, and the unique sequence of bases codes for the unique sequence of amino acids in the polypeptide or protein. And it's the unique combination of proteins that produces a unique phenotype. All right, so again, that's basically what's shown in the diagram at the bottom, and that's what's known as the central dogma of biology. It's the essence of life. So go ahead and pause it and fill in these blanks as best you can, because this is a review. You should be able to either remember or look back in your notes to fill in all of these blanks. So go ahead and pause it and do that, and then we'll fill it in together. Okay, I assume you're ready. So DNA stands for deoxyribonucleic acid, DNA. The shape of DNA? A double helix. DNA is composed of repeating monomers called nucleotides. The 2 polynucleotides of DNA are held together by hydrogen bonds, and again, that's significant because they're weak, and that allows the polynucleotides to unzip, which is crucial for their function. The rails of the DNA ladder are deoxyribose and phosphate, and I abbreviated phosphate here because I couldn't fit it on the slide. Or I didn't abbreviate it, I used the molecular formula. Phosphate is PO4. It's a small molecule of phosphorus and 4 oxygens. 4 bases make up the rungs of the DNA ladder, and those 4 bases are adenine, thymine, guanine, cytosine, and that is the order you want to remember them, ATGC. So you remember that adenine and thymine bind together, and guanine and cytosine bind together according to the base pairing rule. Adenine always pairs with thymine, guanine always pairs with cytosine. The sequence complementary to AATGCA, you should have written in underneath here, and if you didn't, go ahead and pause before I pull them up, but by now you should be able to do this without even hardly thinking about it. A matches up with T, T matches up with A, G matches up with C, C with G. The genetic information is coded in the sequence of A's and T's and G's and C's. That's what spells out the information to make every living thing on Earth.